everyone, welcome to Biomix Sub. In this video, we are going to discuss about the Agrobacterium mediated transformation in plants. So what is this Agrobacterium? It is a gram-negative bacteria. It is a gram-negative bacteria which is present in the soil. This bacteria is present in the soil. Okay, and it is responsible for causing some of the diseases in plants, mostly in the dicot plants. It is not seen in the monocot plants, only it is seen in the dicot plants. So there are two types of species which are present. They are the Agrobacterium tumefaciens. First one is our Agrobacterium tumefaciens, and another is Agrobacterium rhizogenes. As these are the bacteria, there is a extra chromosomal genes which is present, or extra chromosomal material is present except the chromosome, that is known as plasmid, and this plasmid is responsible for causing some of the diseases. So the Agrobacterium mediator Ti plasmid, this tumefaciens is having Ti plasmid and this rhizogene is having Ri plasmid. Okay, and these are the tumor induced genes, and the Agrobacterium tumefaciens is responsible for crown gall disease. Crown gall disease. This is the example of a crown gall disease, and the rhizogene, Agrobacterium rhizogene, which is having the Ri plasmid in it, it is responsible for hairy root disease. Hairy root disease. Okay, this is an example of Agrobacterium tumefaciens mediated disease, which is known as crown gall disease. This is the one which is present. Okay, this is a disease which has been caused by Agrobacterium tumefaciens, and this is a hairy root disease which is caused by Agrobacterium rhizogene due to the presence of the Ri plasmid. Okay, so what exactly is plasmid? As this is a bacteria, in case of a bacteria, what we have? We are having a chromosomal material as well as there is a extra chromosomal material. This extra chromosomal material is known as plasmid. This is known as plasmid, and this plasmid is responsible for causing this disease. Inside the plasmid there is a tDNA present. Let us have a discussion in that. We can see here by this example. See, this is a picture of Agrobacterium tumefaciens. Here we are having the bacterial chromosome. This is a bacterial chromosome, and besides the bacterial chromosome there is a plasmid which is known as Ti plasmid. This is the Ti plasmid because it is Agrobacterium tumefaciens, and if it would be Agrobacterium rhizogenes, it would have the Ri plasmid. In case of a tDNA, it is having one portion, in case of a Ti plasmid, it is having one portion that is known as tDNA, and this tDNA is transported to the plant. We will have a discussion how it is transported. Let us first see how it is taking place. So when this tDNA is transported inside the plant, there it will cause the wound site. When the wound site is present here, it will cause a disease. You can see here, after the inoculation of this bacteria in the wound region, here it is a wound region present, there is a crown gall disease over here. This is the overall scenario of this Agrobacterium mediator transformation. Now let us see what is present in this Ti plasmid. This whole region is a Ti plasmid. Okay, here there are borders present. Here there is a left border and here it is a right border. Inside the left and right border there are some of the phytohormones which are present, and these phytohormones are also present in case of a plant which are responsible for its cell division. What are they? They are the auxin, cytokinin and opine. These are the three types of phytohormones which are present in this tDNA. Okay, and this tDNA is transported inside the plant which is responsible for causing the disease. The tDNA which is present, it is of 15 to 30 kb in length. 13 kb in length, and the right border and the left border which is present, it is of 25 base pair. This left border is also of 25 base pair. Okay. What is the need of this right border and left border? It is setting a boundary that this is the tDNA and the right border is responsible for the transport. If we eliminate the right border or if there is some of the deletion in the right border, the transformation cannot take place from the Agrobacterium tumefaciens or rhizogenes inside the plant, and there will be no disease formation. Okay. Now, what are the other things which are present in case of a TI plasmid? They are the origin of replication, and there are some of the virulence gene. Why there is a origin of replication? As we know that the plasmid is having the self-replicating power. If there is having a self-replicating power, they should have some origin of replication. So this is the ORI reason, which is known as origin of replication. Except that there is a virulence reason. What does this virulence reason does? These are some of the genes which are helping in the transport of the tDNA from the bacteria to the plants. So how this transformation is taking place? This transformation is taking place through the conjugation method. See, suppose here we are having a bacteria. Inside the bacteria there is a tDNA present, or the TI plasmid is present, inside which the tDNA is present. This needs to be transported into the plant. So this is been done by through the process of conjugation. This is the plant shell, and through a pipe like structure the conjugation process will be taking place. From here it will be transported into the plant shell, and inside the plant cell, in the chromosome of the plant shell, it will be ligated through homologous recombination. Okay. Here are some of the genes which are responsible for the formation or for the synthesis of the auxin, cytokinin and opine. Here we are for the synthesis of opine. For the synthesis of opine, we are having OCS or octapine synthase, NOS which is known as nopalin synthase. Next is our FRS which is known as fructopine synthase, MAS manopine synthase, AGS which is known as agropine synthase. These genes are responsible for formation or the synthesis of opine. Next is our auxin. It requires auxin. For the synthesis of auxin, that there is TMS1 gene, TMS2 gene. Next is our phytohormone which is known as cytokinin. For the synthesis of cytokinin, that requires TMR and TML. Okay, the exact function of TML is still unknown. So these are all some of the genes which are responsible for causing the disease in the plants. So in this slide we are having the steps which are required for the transformation of the tDNA from the bacteria or the Agrobacterium tumefaciens inside the plant cell. There are some of the steps which are required for the transformation of Agrobacterium into the plant cell. So what are they? They are the plant stress condition, or if the plant is suffering from some of the wound or injury, it releases some of the phenolic compound. The second step will be the release of the phenolic compound. This phenolic compound is sensed by the bacteria. The third is that signal to bacteria. What happens at first? There is some of the stress condition which the plant is producing, or due to some of the wound which has been produced. Okay, when the wound is produced, they release some of the phenolic products, and these phenolic products are sensed by the bacteria. After the bacteria is sensing the phenolics, then what it does? It helps in activation of the virulence gene. See here, we are activating, there has been activation of the virulence gene. After the virulence gene has been activated, the tDNA which was present in the TI plasmid, it is now ready for transformation. Now there is generation of tDNA. Next is the tDNA transfer. After the tDNA has been transferred, it needs to be integrated into the plant genome. So let us see in very detailed way how this has been done. So as we have discussed earlier, this is the structure of the tDNA. So what is required? We have to see what are the things which are required for this transformation. The things which are required are the borders, the left border and the right border, as well as the tDNA. So the first thing which is required, it requires tDNA. The second thing is that it requires the border region for the transfer. Okay, so next is the third is the virulence region. Third is the virulence region, which is required for the transformation, or we can say it as it is a virulence genes. So what are the virulence gene? The virulence gene can be Vir A, B, C, D, E, F and G. These all are the virulence gene. They are responsible for various type of functions through the for the transformation of this tDNA. The next fourth one is that it requires a chromosomal virulence gene. Virulence gene. So in details we will discuss what are the function of this all Vir genes. Okay. So here we can see that here the plant is suffering from some of the wounds. Here the plant is suffering from some of the wounds. That means injury has been taking place. There is formation of, during this time there is formation of some of the phenolic compound. So what is the phenolic compound which is produced? That is acetosyringone. This acetosyringone is a phenolic compound which is produced by the plants, and this has been sensed by the bacteria or the Agrobacterium tumefaciens. It has been sensed. What happens in the next case? There has been activation of some of the virulence gene during this time. So now there is one question that if this phenolic compound, that is the acetosyringone, has been released, so when the bacteria is sensing it, there must be some of the receptor which is recognizing the acetosyringone. So which is that receptor? They are the Vir genes. Of that, Vir A is responsible for identifying that there is a syringone, we have to receive it. So here we are having a diagrammatic representation how the Vir genes are responsible for the transfer. So we have discussed there are various types of gene from Vir A, B, C, D, E, F, G. Okay. So whenever there is a bacteria, here we are drawing the bacteria. So what happens in the first case? This plant is producing a wound. When the wound is produced, it releases a phenolic compound. What is the name of the phenolic compound? It is acetosyringone. This acetosyringone is received by a receptor which is known as Vir A, which is present in the bacteria. It is a sensor kinase. That is the reason it's sensing that acetosyringone is present now, so we have to receive it. What happens as it is a kinase, what it does, it is having a autophosphorylation activity. After the acetosyringone has been reached here, it is phosphorylating itself. In which region it is phosphorylating? It is phosphorylating in the histidine amino acid. Histidine amino acid, and it has been activated. After that, what it does, it transfers its phosphate group into Vir G. This Vir A and Vir G are constitutively expressed in this cell. So after this has been activated, there are activations of some of the other genes. So what are the other genes? Vir B, C, D, E, F and G has already completed its work. So let us see how it works. In this slide, we will discuss the various type of Vir genes and its function. There are some of the virulence gene which is responsible for the transfer of the tDNA into the plant shell. So there are various type of Vir genes like Vir A, B, C, D, E, F, G. So what is the main work of Vir A? The main work of Vir A is that when the plant is suffering from some of the wound or under stress condition, when the acetosyringone, which is a phenolic compound, is present, it will receive it. Suppose this is a bacterial cell. And Vir A is present over here. Why it is present? To receive the phenolic compound which has been produced by the plants. This has been received by Vir A. And as it is a sensor kinase, it is having the autophosphorylation activity. And the phosphorylation is taking place in the histidine amino acid. And it has been activated now. After the phosphate, this phosphate will not remain with it. It will transfer it to Vir G. Here it will transfer it to Vir G. Okay. In the Vir G, it will transfer the phosphate group into the amino acid as per Tate. Right? After this 2 has been activated, see here it has been written that weed gene activates other weed G activates other weed genes. They are like the transcription factors. There are weed B, weed C, D, F, H, F. This needs to be activated now. How this will be activated? Now, there is some of the genes which are responsible for formation of the cut, which is that they are the weed D. Weed D is responsible for the cleavage, where the cleavage is needed. We have already discussed. What was the tDNA, what is the Ti plasmid. So, now we are discussing about the weed D1 and D2. The weed D2 which is present, it is responsible for the formation of the cut because it is having the endonuclease activity. Here near the right region and the left region, the weed D2 will come and cut, and it will be transported here. When there is formation of the cut, this weed D2 will remain with it. Suppose we say this is the enzyme or the weed D2, it will remain until it reaches a plant, and the right border is responsible for the cut. It recognizes the right border for the formation of the cut for its endonuclease activity to be done. And the weed D1 which is present, it is having the topoisomerase activity which is guiding the weed D2 to do its work. Okay. Now, next comes our another weed gene which is known as weed E. The weed E which was present, there was weed E1 and weed E2. This weed E1 is stabilizing the work of weed E2. Okay, so what is the function of the weed E2? When this E2 is present, it will bind with this DNA. This D2 will bind its DNA, and when this is transported, it will not be transported in the double stranded form, it will be transported by the single stranded form. Okay, and this weed E2 is working as a single stranded binding protein. In all part of the DNA, this E2 will be bounded. Why it will be bounded? To prevent it from recoiling or self-replicating in the meanwhile. Okay. Now, what is the next work? The next work is of weed B and weed D. What is the function of weed B and weed D? It is responsible for the formation of T4SS channel. Okay, let us see what is this channel. Here we are having a channel. Let us see here. Here we are having a channel-like structure which is responsible for the transfer, and this channel is being done by weed B1 to B11 and weed D4. It is responsible for the formation of this channel. Why it is known as T4? Because it is known as type 4 secretion system. Okay, this channel is known as type 4 secretion system, and for the formation weed B1 to weed 11 and weed D4 is responsible. So the next function is of weed H. Weed H. Weed H is host specificity. And the weed C12 and weed C2 is enhancing the work of weed D1. Okay. Let us have a discuss write all the words of the weed genes which are taking part in the Agrobacterium mediated transformation. So the first verse is weed A. The weed A was working as a protein kinase which was responsible for sensing the phenolic compound that is known as acetylserinone. The next one was a weed G. Weed G. Weed G was helping other weed genes to get activated. The next one was VD1, VD1 and VD2. The function of VD2 was endonuclease. Endonuclease, that means it is cutting near the right border and it is attached to the right border till it is being transported. And the work of VD1 is help D2 is a function of VD1. Next come the Vir-E. The Vir-E is working as a single stranded binding protein. It is binding to the DNA. Next comes the Vir-E1. The function of Vir-E1 is that it helps Vir-E2. Vir-E2. Here the single stranded binding protein is a function of Vir-E2. Now the work of Vir-C1. Vir-C2 to C12. What does it? It enhances the work of Vir-D1. Next is Vir-H. Vir-H is host specificity. It is host specificity. Okay. Now the last is Vir-B2, Vir-B1, Vir-11 and Vir-D4. The work is T4 secretion channel. These are responsible for the formation of T4 secretion channel. This is all about the Agrobacterium mediated transformation in plants. Thank you for watching this video till the end, and if you like this video, please do like, share and subscribe. Thank you.